I'd like to move on to Dr. Kenyon Mobley, who, so Kenyon, if you want to share your screen, that would be fantastic. And
can you see my notes? Sorry.
no, you've got the right screen up. That looks good. And Kenyon, Dr. Mobley will be, who's been a long-time active researcher in signathid biology and evolution, is going to talk about snake pipefish in the northeastern Atlantic Ocean and the boom and bust in a panmictic population. Kenyon, you have 20 minutes.
Great, thanks, Healy, and greetings from the north. I'd like to start my talk today by thanking the organizers for putting together this truly international conference and continuing the tradition of promoting signathid biology and conservation efforts. I'm really pleased that the tradition continues. Today, I'd like to talk a bit about a study that I started during my PhD on snake pipefish. Wasn't part of my PhD. Let's see, can we go on? There we go, okay. So the snake pipefish is a rather large European pipefish, and you may recognize it from the first 3 Sing biomeeting logos. Enteloris are one of the few species of signathids that can occupy ocean habitats and can be caught at up to 100 meters depth. Records, mostly based on museum species, describe them as having 2 forms or ecomorphs, a coastal or a littoral form, and a truly oceanic form. They aren't considered particularly strong swimmers. They do have a slightly prehensile tail. It's very long. They're often found among floating wrack in the open ocean or in seaweed along the coast and even rocky shores. Okay, now this snake pipefish is commonly found in temperate to subarctic waters along the continental shelf in the northeastern Atlantic Ocean. They're found as far south as the Azores to about midway in Norway in the north, and they can go all the way out to the mid-Atlantic Oceanic ridge. So their range is quite impressive. The highest abundances are found, though, in the North Sea and the Celtic Seas, which I've outlined in red.
If you are not speaking, could you please mute so that we can let Dr. Mobley speak? Thank you very much.
Thanks, Healy. I'll continue. It scared me a little bit. Okay, and so they're basically found around the British Isles and even up to Iceland. We find lower abundances of them in the Bay of Biscayne, along the French coast, the Portuguese coast, the Spanish coast, and there's even records of them perhaps in the Mediterranean, but it's unclear if these are misidentifications, occasionals, or true populations. So if anybody is listening that has some information about these populations in the Mediterranean, I'd be very interested in talking with you. They're also found in the White Sea in Russia, and this could be some sort of relic population. I've not been able to get samples from this area yet. But what I wanted to tell you about today is that during the years of 2005 and 2007, Enteloris, the snake pipefish, underwent a massive population explosion. And they were found all over the North Sea and on the continental shelf in very high abundances. So normally we find, maybe a few per sample, but here they're showing up in all trawls and in enormous numbers. And even in Sweden, where I was working in 2003 and 2005, I caught a whole bunch towards the end of summer. I didn't think much of it because I hadn't been there that late in the year, but it turned out that it was the beginning of what we now consider a large population explosion. They also went through a range expansion, although you might question whether or not it's actually an expansion if those populations never really colonized. But they were found as far north during this period of time as the Greenland and Barents Seas and the Kola Peninsula, which is just kind of outside the right-hand corner of the map there, so around Norway and the north part of Russia. They appeared to have been rafting, and no reports of these populations have been reported, seeing whether or not they've been established or not. So again, I'm interested to know if anybody has any information on this. But during this time, there was quite some concern about ecosystem disturbance due to this very high abundance of the species. So this is really the reverse of what we saw this morning about looking at conservation. This is what we have too many of them, okay? And for example, some of these species really seem to enjoy the pipefish and made nests out of their dead carcasses. That's what this black-legged kittiwake is doing. If you can look really closely at your screen, that is completely comprised of Entelurus dried carcasses. However, the downside to this is that because, or rather potentially due to the fact that there's so many pipefishes out there, they started feeding their young these pipefishes, which have all the nutritional value of, I don't know, like some Legos. And so this is known as the junk food hypothesis, and seabirds were suffering greatly during this period of time. So there was a flurry of papers that were coming out, but we didn't really know much about the basic biology. And as it now stands, the causes for the sudden increase in abundance and the subsequent decline, starting around 2008, this decline, are unknown. However, the most likely culprit is sea surface temperatures. And during 2005 to 2007, particularly in the North Sea and the Celtic Seas, the temperature was a couple of degrees higher, about 2 degrees higher than what is normally experienced. And so what I'm showing you here are some sea surface temperature reports from week 36, which is around, it's on the cusp of between August and September. The temperatures around the North Sea and the Celtic Sea, around like the bottom part of England and Ireland, they're in orange, but when we get to 2010, we see this is firmly in the yellow place. So the temperatures, and this is a trend that was seen all over the place. And so this could have had something to do with why they were doing so well, because there was associated phytoplankton and zooplankton abundances and changes in these particular ecosystem, and perhaps even the temperature is more favorable to the reproduction. And so some other possible explanations include there's been some decadal shifts in phytoplankton abundance in particular, and also an invasion of southern copepod species, which are likely tied to the sea surface temperatures. And there's also the potential for a decrease in competition for food, which may have either been correlated or as a result of an increase in the snake pipefish. So for example, people were recording that there was low sand eel abundance in these areas in the North Sea, which may have led to the pipefish having way more food to eat. Okay, and so during the summer of 2005, I collected quite a few Entelurus in Sweden, and I clipped a piece of their tail and kept it in some paper bags for some later genetic analysis. And my original goal was to develop some microsatellites and do some parentage analysis more akin to what I was doing during my PhD. However, when these studies started creeping up a few years later concerning the population boom, I realized we knew so little about the species that I thought it would be interesting to go back and investigate the phylogeographic history of the species. And so, for example, what I wanted to do was to ask, was there even a population structure? There were rumors that there was southern populations invading the north, or this oceanic species was invading the coastal. And well, that all is hearsay unless we can actually go and look at this more scientifically. And there is also whether or not there's these genetically distinct, what I'm calling ecomorphs in In the literature, they use other terms, but ecomorphs in the sense that they have two different phenotypes for these two different environments. And whether or not we can detect any historical population expansions or signatures of glaciation, which has affected many of the populations or species of sygnathids, particularly in the temperate areas. So in 2010, together with my colleague Inez Braga-Gonzalez, we contacted as many researchers and museum collections as we could to get samples of Entellurus. We aimed to cover as much of the distribution as possible with their samples, and we got samples as far south as the Spanish coast from Tony Wilson. We got some small funding to go to France, which is shown in blue, this French coast, and Roscoff to collect Entellurus there. But the bulk of the samples, and there's also some museum collections shown in orange, and then my collections in Sweden as shown in orange. But the bulk of the samples came from two collaborators in the Netherlands, which is Bram Couperus and Cindy Van Dam, who had huge amounts of fish that they got from these Necton trawls out at sea. And I spent one day there going through all these massive bags of frozen fish with Bram over lots of cups of coffee, and got as almost, I think altogether with all of our collections, 178 samples from 25 locations spanning this 8 years. So kind of during the population explosion and then after the population explosion. And we assigned these, as you can see from the map, we assigned these samples to 6 different geographic areas, and this corresponds roughly to their physical location. We sequenced, we sequenced 120, 1,200 base pairs of the cytochrome B, which is a mitochondrial DNA marker, and about 600 base pairs from the control region, also a mitochondrial marker. And these are traditional markers for phylogenetic studies that generally show patterns of population subdivision and glacial influence in several signated species. So to cut to the chase, what we found using these mitochondrial markers was well, first I'm showing you the haplotype networks on the right-hand side of cytochrome B's on the top and the control regions below it. What we found is 4 large haplotype groups that are fairly closely linked. But what was interesting to note is that there was, based on our population or our region areas, we found those haplotypes occurring in virtually every area that we sampled. So what we would expect is that these guys have fairly high gene flow between these different areas, and that there's no particular haplotype that demarks particular locations. So there's no southern or northern populations. There's no strong evidence for this, at least with the markers that we have. We also found high haplotype diversity and low nucleotide diversity, and these are characteristics of a fairly recent expansion. We also found really no evidence for any coastal or oceanic population structures. So we don't believe that that's real, at least at the molecular level. It could be phenotypic, but I'll discuss that in a little bit. We also investigated patterns of population expansion using both mismatch analysis and a couple of other things, as well as some Bayesian methods. And shown on the right is the skyline plot showing the changes in effective population size, so the number of breeding adults through time based on our mitochondrial markers. And this is the combined cytochrome B and control region thing. It's a bit dependent on your mutation rates, and so we played around with this quite a bit, but what we basically found was no matter what we did, we were finding a fairly stable population size for about the past 50,000 years, and then there was some expansion before that. Now, this is interesting because the most recent glacial activity is about 10 to 20,000 years ago, which most other signathids show. We don't see that. It looks like it's much earlier than that. And when we originally tried to publish this, we got a desk reject from a very famous biologist who said, if you don't have any glacial signature, then something's wrong. And I was like, well, what if that's actually right? I mean, isn't that what science is? I mean, he was basically trying to force us to make the glaciers be as kind of a yardstick in order to do this. But in any case, what we think could be happening is that because of their oceanic lifestyle, they may have shifted their range and avoided some major impacts of the recent glacial activity, or had a population so big that there isn't really any evidence of this, right? So one of the things also when we were going through this is we wanted to see whether or not there's any historical evidence for other population booms of the snake pipefish. And we came across this this woman's work, Margaretha Bongerisma Saunders, another famous Dutch scientist, and she made it her life's work to study mass mortality in the sea. And she noted that from ship's logs from starting at 1855, that there was mass amounts of snake pipefishes dying, and these were usually found by the mid-oceanic ridge, which the first person that saw this, they attributed to volcanism, but there wasn't any other recordings of volcanism in this area. So, you know, perhaps not. This is a long time ago. There was another expedition by the Prince of Monaco in 1887, who also noted huge die-offs of snake pipefish, and they thought that they were commensals with salps, which are colonial tunicates, but that might be just because they were all wrapped up in the same kind of drifting mass. By the way, all of these records are close to the mid-oceanic ridge, which is right on the edge of their range, which might be that they've reached an area where they just can't take it anymore and they die off. But this means that there's huge amounts of these guys to begin with for a sailor to note, or a captain to note, that they're actually washing up. And finally, in 1911, there was another record of this mass die-off, and there they made the suggestion that salps and microplankton blooms were killing them, even trying to eat them and drag them across the water. I mean, this is interesting, but I don't think that there is much basis for this. In any case, to summarize, based on the evidence that I'm presenting you today, we believe that this is one large single panmictic population of snake pipefish in the Northeast Atlantic Ocean, with significant gene flow across over 2 million square kilometers, at least over the samples that I collected. We think this high level of gene flow is a combination of their pelagic lifestyle, perhaps fast development times, and long distance dispersal aided by ocean currents. We also didn't see any evidence for genetic subdivision among these coastal and oceanic populations. And going back to some original descriptions by Dunker in 1915, these designations of this coastal and I forget the other term that he used, coastal and oceanic populations, these were all based on dead individuals. And so the question is whether or not some of these kind of, you know, other ecomorphs could have been just dead individuals that look like they're in particularly poor condition. Another potential issue is that he may have been classifying females and males wrong. Who knows? I don't know. In any case, we did not see a range expansion as establishing, but they certainly did get up far north. So we think this is rafting. And the range expansion in the Pleistocene much greater than, or much earlier than what we would expect for other signathid species, suggests that this population has been stable for a long period of time in the Atlantic Ocean. Right. So going forward, I have to say that this was always a project of love, and we never received significant amount of money to do this kind of work. So this was always kind of slipping this, you know, some stuff on plates that I was running for other things. And I hope nobody's listening to this talk that had something to do with that. In any case, I would love to go back and do something with improved genomic resources. I mean, we can now, for what I could have started 10 years ago, we could have probably gotten some decent markers, some SNPs, something like that. I still have all the samples too, although they might be somewhat degraded. And also improve a network for collection of samples. I mean, we are not out of the woods. It could be that we have another warm period and another large population of snake pipefish explosion. So it would be, I'm going to try harder to get some more samples from folks if this happens again. In any case, I thank you all for the time. There's many people that helped me out, collect and donated DNA samples for this project. But mostly I want to thank Inej Braga-Gonzalez. She's shown here on our trip, our fateful trip to France, where we caught all of our fish in one day. And Luca Coronetti, another co-author who performed the Bayesian analysis. And of course, Bram Kupris and Cindy Van Dam, who supplied the bulk of the samples. And yeah, I think that's it. We did get some funding, but it's mostly for personal salaries and the fated trip, which was very nice, to France. So thank you very much.
Thank you very much, Kenyon. And of course, you're getting all the electronic applause that you can't hear, but it's there. So we are running a little bit behind time, and I did ask for questions to go in the chat. While we're moving over to Vasily's talk, we could look for one question. And you can also feel free to put a question in the chat that Kenyon can answer. And if there are not any immediate questions, I'll just say I think it's super interesting because back to the phylogeny, Entellurus is very basal, and so I just kind of wonder about some of the primitive characters that it has, and especially that pelagic dispersal, just what that how that influences the evolution of the rest of the group.